allow people in from the waiting room, and then we'll get started.
Recording has started to the cloud.
Good morning, everyone. My name is Othira Ginsberg, and I'm the Senior Advisor for Clinical Research at the U.S. National Cancer Institute Center for Global Health. Thank you so much for joining us today for our Global Cancer Research and Control Seminar Series. This seminar series features talks by researchers and cancer control experts working in global oncology and provides opportunities for discussion and collaboration on impactful and innovative work addressing cancer morbidity and mortality worldwide. I'm delighted to welcome today Drs. Gaurav Narula and Hasmukh Jain to present their work in the development and initiation of India's first CAR T cell therapy program. In today's seminar, Drs. Narula and Jain will cover their early collaboration with the National Cancer Institute, development and results of their pediatric and adult clinical trials, and commercial approval and rollout of their program. Also Surgeon Commander Gaurav Narula is a professor of pediatric oncology and health sciences at Tata Memorial Center and Homi Bhabha National Institute in Mumbai. A veteran of the Indian Navy, Dr. Narula served for 25 years as a medical officer in various naval ships and hospitals. Dr. Narula's work focuses on immunotherapy and collaborative clinical trials for acute lymphoblastic leukemia, ALL, Hodgkin lymphoma, and histiocytosis. Since 2015, Dr. Narula has been collaborating with the Indian Institute of Technology, Bombay, to develop the first CAR T product in India. Dr. Hasmukh Jain is a medical oncologist in the adult hematolymphoid and cell therapy unit at Tata Memorial Hospital. Dr. Jain received an MD in general medicine from Kasturba Medical College and DM in medical oncology from Tata Memorial Center. Dr. Jain's primary research focus is ALL, Hodgkin lymphoma, cell therapy, and supportive care. He served as PI in the clinical trials that led to the approval of the first CAR T cell therapy product in India and is now working on ways to improve access of CAR T cell therapy to Indian patients. Now, before handing the session over to our esteemed speakers, I'd like to review a few logistics. First, please save your questions for the designated Q&A time that follows immediately following the presentation. And you can raise your question in the chat box or use the raise hand feature found under reactions. And we'd actually like people to put themselves on video if you feel comfortable doing that, if you're called upon to ask your question. Also note that today's presentation is being recorded and will be available on our website at cancer.gov/global health. Finally, you can find information about future presentations on our website, cancer.gov/global health, and by following us on X at NCI Global Health. And now we'll begin our presentation. Welcome, Drs. Narula and Jain. And please note also, during the Q&A, we'll have their colleague, Dr. Rahul Purwar, available also to respond to questions. Thanks so much. Please, the floor is yours.
Thank you very much, Ophira, for that kind introduction, and thank you very much to NCI for inviting us to share our journey. And it's only right that it should be here in this forum because this is effectively where it all began, and I hope to cover some of that aspect during my talk in the next 20 minutes or so. So, as you've already been told, I'm from the Tata Memorial Hospital, and that's the bunch of buildings in the center of the screen over here. And this photograph was taken just after a cyclone had passed the city in 2020. These are my disclosures. So really for us, the CAR T cell journey began, for me personally, and I'm just taking you through a slightly personal story with this over here. With this email which a colleague of mine had sent to me just shortly after I'd left the Navy and joined Tata Memorial Hospital. And when I opened the link, it led to this story of Emily Whitehead, which I think a lot of the world is familiar with by now, who was on her last legs for ALL treatment, and nothing was really working, and she was the first child to receive this CAR T cell therapy. had a very dramatic course, and she had very severe cytokine release syndrome, and then she came out of it because they serendipitously discovered that tocilizumab might work for her. And of course, when I discussed this with my friend, I cannot forget his words. He said, Gaurav, this is science fiction. And this was way back in 2012. But in 2012, when I joined Tata Memorial Hospital, we were facing different challenges. And this is a standard, slide which we show in all our pediatric ALL discussions. ALL is, childhood ALL especially, is a success story of cancer actually. And this graph of successive COG trials, Children Oncology Group in the US, shows you how outcomes have been improving over the decades. And from a nearly completely incurable disease in the 60s, it's now about 90 to 95% of children can be cured long-term of this disease. And this was the status in 2006. We are not even reached the 2020s. But in developing countries like ours, the story is very different, and outcomes hover around the 70 to 80% mark. And that is also now with a lot of changes. So in 2012, when I joined, I took this cohort, and we took patients who had started treatment in 2010 and followed them up, and we saw, sure enough, 65 to 70% overall survival was what we were getting. So before getting to CAR-T, it's always important, because I get asked this question a lot, that first we need to improve our basic treatment, and we have been doing that, at least in our unit and across the country. Similar efforts have been there. We put into place a very comprehensive social support program, ensuring that our children could get financial support, accommodation, good nutrition. A lot of them come malnourished, and even adequate platelets and transfusion support. And we've even started making efforts to continue their education and vocational training while they were undergoing treatment with us. And this is Shalini Jatia, this wonderful lady, who leads these efforts for us through our foundation at the Tata Memorial Hospital called Impact Foundation. Well, this Impact Foundation had its impact. And just to get you an idea of the kind of workload we do here and why CAR-T is so imperative to us, in 2010, as you can see, we had 600 new cases registering in my unit. This is the pediatric hematolymphoid unit. We see only children with leukemias and lymphomas below 15 years of age. And by 2018-19, we were crossing 1,000 patients a year. It's only during the pandemic and the lockdowns, because of travel restrictions, that it dipped, and it's bounced back right now where it was. The graph on the right shows you how our funding situation improved over time, and now we can ensure that every child who comes to us, regardless of economic status, can get complete full treatment, and this is very important. And you can see these graphs on the bottom right, which show you how the treatment refusal and abandonment, which was nearly 20 to 30% in the late 2000s, has dramatically come down with the availability of funding and our comprehensive support program, down to less than 5% consistently for the last several years. And this is huge for a country like ours, where most of the patients, or maybe half of them, can't afford the treatment, and another third out of those who can are going to become probably devastatingly poor by the time they're halfway through treatment. So having put this in place, we felt its impact, and these two lines show you the earlier era, where outcomes were around 60%, and which has now started improving to 70 to 80%. But this was also due to changes in our protocol, using more modern stratified response adaptive protocols, and that caused improvement. But when I started looking at the outcomes of these children who were relapsing or who were refractory, they were dismal. So the frontline treatment was improving, but the relapsed refractory treatment did not move. And the reason for that is that, most of these children required a transplant, and now we have had transplant for 4 decades. In fact, in 1983, our hospital, Tata Memorial Hospital, pioneered the first transplant in India, and we still are amongst the oldest and amongst the larger, transplant units in the country. But the wait list was reaching nearly a year, which meant the children who needed it the most could not get it. So we started looking at CAR-T not as a bridge of, the final 5% of outcomes which the West is seeing, the final 95%, and that the graph which I showed you is before CAR-Ts came into the picture. In the West, you're trying to bridge that last 5%. We want to jump from 70 to 80% to 100%. Even if we triple our transplant capacity, we are not going to get there. We need something more than it. And that's what, really drove our passion and our need for this new novel therapy. And this started as a collaborative effort, and this is I'll cover that in a bit. It was Dr. Rahul Purwar, the scientist from IIT Bombay, who will also be in this panel and available for Q&A later on. We sort of met, and I'll cover that in a bit, and we sort of hashed this plot, maybe if you would like to call it that. A lot of the preclinical work was done in Dr. Rahul's lab in IIT Bombay, which is a premier technology institute of our country. Translational work, etc., and other things we worked on together. The clinical trials were started in Tata Memorial Hospital. Helping us through this entire process was a National Cancer Institute, and especially I must acknowledge the contribution of Terry Fry, Dr. Nirali Shah, and Stephen Heifel, amongst many others, and the Center for Global Health, which helped to coordinate this. So this is the timeline. From beginning to end, we did not receive any financial or material aid from NCI. It was literally mentoring and knowledge partners. I think these three especially, and many others, just opened their minds for us to pick at any time, and we made several visits across, which helped us in this process. So these are the milestones that we went through in 2015, and I may just take a moment to elaborate on this a bit. It really started with an accidental meeting with Terry Fry. Dr. Terry Fry was part of an NCI delegation to Tata Memorial Hospital and other centers in India in the February of 2015. But he had no official engagement in Tata, and we just got a phone call saying, there's a delegation from NCI, but there's a pediatric oncologist and transplanter here, and he's free, he doesn't have any engagements. Would you guys like to interact with him? And when I was told that, I said, my God, Terry is here. Of course, we want to meet him. So we arranged a talk for him in our department, and then we interacted with him. And I pitched this idea to him that, this is what we are thinking of. And it was very early stage for CAR-T at that time. And he said, yes, okay, we can help you out. And he connected me to the Center for Global Health and Sudha, Shivraman, and others over there. And this is really, and Dr. Vidya Vedam and others. And that's what really got us started. In May, I met Dr. Rahul Purwar and discovered that he was already working on CAR-T right in our own backyard. And it was after seeing that work, we decided we could work together, and that's the collaboration which I told you about. Well, moving forward, we started formalizing our associations with NCI and with IIT Bombay and amongst each other. And we started trying to apply for grants. This was a very disappointing period where everything we did was rejected. I don't think anybody, really thought we could seriously do it. Our institutions backed us to the hilt, I must say, Tata Memorial Hospital and IIT Bombay. And with small grants, we kept going till in June 2017, we got a sort of grant from Tata Trusts, which sort of got us going a bit more. And by October 18, the products had been developed in Rahul's lab, and preclinical work was completed. And then we started working on the manufacturing and regulatory parts. And it was in March 21 that we finally got approval for commencing the first phase 1 trials in India. It was a historic approval because it's the first time it was being done in our country. And in June 21, the first patient was infused. By December 22, we completed the phase 1 trials and started phase 2. And by October 23, as a result of the phase 2 trials, the adult patients on lymphoma and leukemia, which was done by Hasmukh, will be speaking subsequently, a very landmark historic trial done over here in record time that we got approval for use in adults. And that, in a nutshell, is how the whole journey went. There was a short-term scientist exchange program set up with the NCI through the Center for Global Health, which allowed us to visit NCI several times. Myself, Hasmukh, Dr. Rahul, we all visited, and including other members of our teams, including PhD students and postdocs. And also members from NCI came, including Dr. Terry Fry, Nirali, and Steve, all visited India, and especially also from the Department of Offices and Spaces, who helped us with our GMP and manufacturing sites, design, and which was something which was totally new to us. So now I'll just take you through a little bit of the preclinical work which led to the approvals. So this is Dr. Rahul developed in his lab 3 new novel constructs. 2 were humanized, one was murine, and based upon the affinity data, we chose to go ahead with one of them called H1CAR19 and proceeded with this because it was showing the most promising data. Here's a little bit more of the clinical data. This graph shows you that, we could, the cartoon on the left shows you that we could consistently get good transcription efficiency. And this work was done by Alka Dwivedi, and who was Dr. Rahul's PhD student, and Arthav Karolkar, who's also another brilliant guy. Alka is now in the National Cancer Institute in Bethesda as a postdoc, and Arthav is also with Dr. Rahul in ImmunoAct, and very bright students who did this work forward. This shows the efficacy in various cell lines, and it showed a very good efficacy, especially at a 5:1 ratio, and no effect on the CD19 negative cell lines. And this is a bit interesting for those into the CAR T space. We found that the cytokine production was fairly low, and this we thought was good, especially when it came to TNF alpha. We thought that would produce less CRS, but it was a bit concerning at that time in the 2016, 17, 18, when we found interferon gamma production to be low. We thought this might affect efficacy, but we were seeing good, robust activity. So we could not sort of tie this in, but we went ahead with our gut feeling that it was efficacious. So fortunately, later on, before we got, there was literature coming out that interferon gamma is more important for solid tumor CARs, but in hematological malignancies, it did not seem to matter that much, which sort of corroborated our feeling, and we went ahead with this. The novel construct developed over there in IIT Bombay in Rahul's lab had met several claims, more than 50 each, for novelty, inventor step, and industrial applicability, was patented and had a freedom to operate. And this shows you the animal data, which when we took not-scared mice and irradiated them to knock them out further, gave them a cancer cell line and the CAR T the next day. And these mice, which got no T cells or untransduced T cells, went on to develop full-blown disease, whereas those which got the CAR and the two different doses of the humanized CAR and the murine CAR did not. So with this, we were able to go to the next step, which was manufacturing, which was something we had to do ourselves. There was manufacturing is, as you know, done by Big Pharma, but this is the first time we were manufacturing something inside an academic center. So Dr. Rahul got his own office converted into a GMP center, and this was lucky. Thanks to a lot of interactions with NCI, we were able to pass the norms and get approval from the drug controller, which is our FDA, to use this facility for the early phase 1 trials. So literally, you can say that our CAR originated in a garage. And in the Tata Memorial Center, parallelly, we built a GMP center, which was funded through academic grants. And with this, the aim of this GMP center was to enhance capacity with the CAR T production, but also multiple cell therapy processes for the R&D and early stage development and small scale early phase clinical trials. And then the next challenge was the regulatory network, because we have an advanced and very comprehensive regulatory network. In fact, a very thorough regulatory network existing in our country. But there was no way forward for cell and gene therapies at the time when we had started. So we benchmarked ourselves to US FDA requirements. And as we went along, the committees were literally the guidelines were evolving as our project moved. Actually, many of these committees, which you see over here, were handling a cell therapy project for the first time on the table, even as the laws were evolving. But we were fortunate that it was very, while we faced a lot of extra scrutiny because of that, but it also, people were very supportive and helped guide us through the path. With all this, the preclinical work was published, the clinical trials commenced, and 4th June 21, we infused the first child in India, who was a case of relapsed refractory ALL in the bone marrow transplant unit in Tata Memorial Hospital. And less than 2 weeks later, Dr. Hasmukh Jain, who you see in the bottom right, along with Rahul and others, started the adult trials in lymphoma patients, phase 1 trials, and he'll touch on them later. So with this, I will now just briefly walk you through the pediatric trials, which I was leading here in Tata Memorial Center. We started with a pilot trial in which we explored only 2 dose levels, 1 million per kg CAR T cells, and we went up in the second dose level to 3 to 5 million per kg, capping it at 250 million. But this was to be followed by a phase 1B, but we thought we could go higher. So we introduced another dose level in the phase 1B trial of 10 to 15 million per kg, and this was supposed to be a roll-on to phase 2. The trial design, and these trials were registered on our clinical trial registry in India, and the trial design permitted us to analyze both these groups together for all 3 dose levels to find the phase 2 dose level and move forward. So I'll just present to you now the phase 1B, that is, which clubs these 12 patients who were treated at these 3 different dose levels. This is a high-risk cohort of pediatric ALL patients who had a median of nearly 20% blasts at the time of screening and ranged from 0.1 to 88%. This is really high-burden disease. Most of them had very high-risk features. The complex cytogenetics, which you see here, showed that these were very high-risk patients. 3 of them had past CNS disease. The median lines of prior therapy were 3, ranged from 2 to 6, including past transplants in 3 patients. 2 of them had undergone 2 prior allogenic transplants before they landed up in the trial. Almost all of them needed holding therapies because of the aggressive and late stages of the disease which they were in. And because of that, the bone marrow blasts did reduce from the screening till the time of infusion, but still 4 of them had about 40% blasts. The vein to vein time was about 18 to 133 days. That's because there were some outliers. One patient, especially between after collection, developed severe sepsis, came out of it and developed severe COVID. And once he came out of that, only he could be taken on. And he was very sick by that time. So 4 patients received a dose of below 3 million per kg because one who was intended to receive 3 to 5 could not receive it. So we clubbed them together. And 5 patients received 3 to 5 million per kg and 3 patients received 10 million per kg. This is about the toxicities. Peak toxicity was only grade 1 CRS in about half the patients, and only 1 patient went on to develop grade 3 CRS. This is the same patient who had sepsis and COVID prior to CAR-T, had baseline IL-6 levels in the hundreds even before he got his CAR-T, and the same patient had a grade 2 neurotoxicity. And so far, the entire pediatric trials and subsequent patients also remains the only patient to have had neurotoxicity so far. The rest of the toxicities were mostly unremarkable, mostly accepted. Tocilizumab use went up to 42% because later in the trial, we were using it even for grade 1. Coming to the responses, in the entire cohort, you can see that by day 30, we had 92% responses. 11 out of 12 patients responded. Only 1 had no response, but complete responses were 67%, and about 25% were partial responses. None of the partial responses were sustained, but when we divided it into seeing the patients below less than 3 million and above 3 million, those who received above 3 million, we see that the response rates were nearly 100% at this dose level, of which 88% were at complete responses, which were sustained till day 90, and for all those who have crossed day 180 also. This is a swimmer plot which shows you each horizontal bar is an individual patient. The bottom block is the group of patients who received doses below 3 million per kg. While a couple of them did have some responses, they were not sustained. One patient, the first patient, actually had a complete response, but remained in remission. But before 6 months, he was losing his B-cell aplasia, so he was recommended to go for further therapy and a transplant, which he did about 1.5 years down the line. But unfortunately, he nearly 2 years post CAR-T, he had a relapse after transplant and then passed away. But above the 3 to 5 million and 10 million per kg, you can see every patient responded except one who had a partial response, which was not meaningful because he progressed rapidly. But everybody else remains in a sustained remission. But these bars have grown longer by 5 months since they were last made. These graphs show you that the patient's bone marrow disease burden at the extreme left, and with treatment before the time of infusion, most of them came down, and those who came down went on to have deep responses. But those who did not go down had responses, except this one patient, but had responses even down to probably to minimal disease level, but these were not meaningful. CAR-T expansion dynamics on the graph on the right showed good expansion in almost all patients, except this one at the bottom, which you see this green line, which corresponded to the patient who did not have a response. And the cytokine data, this IL-6 was high in only the solitary patient who had COVID going into, and that's what's changing the scale of this graph. But otherwise, you can see that this was a very low cytokine producing very low CRS producing kind of product that we saw. And the same goes for interferon gamma. This is actually, it's scaled to 200, but in higher levels, it should go higher, but we saw very low levels. And even the ones where it was elevated did not correspond with clinical symptomatology. So with this, the landscape has been growing. In 2021, we started 2 phase 1 trials, which started from Tata Memorial Center in IIT Bombay. Then Dr. Rahul Purwar went on to found ImmunoAct, which took over the phase 2 trials. Another company called Immunil started trials in Bangalore. And by 2023, there were 2 more phase 2 trials opened, some from our own group. And CMC Vellore has also opened, and then both these groups started opening in multiple sites. So as you can see, the clinical trial landscape is rapidly lighting up in India. But a sober reminder, this is a graph by 2020 showing the increasing number of clinical trials across the world, which had crossed nearly 1,000, 2,000 plus by the end of 2020. And this is in CAR T-cell alone. And this is to remind us that where we are now is where the world was about a decade ago. So there's still lots to be done. But this graph gives us much more pleasure. Maybe it's the baby steps in the clinical trial landscape, but we now become one of the 11 countries in the world which has an approved CAR T product. And more on that from my colleague, Dr. Hasmukh Jain, and Rahul Purwar will be covering it later. What we could do over this, as a result of this project, was to develop an ecosystem of team science in the cell therapy space, from R&D through translation, manufacturing, trials, commercialization. And now we are working on access to ensure that even our poor patients will be able to get this. And maybe I'll cover that in detail in some other session, or not, it's not in the purview of this one. And with that, we are proceeding with collaborations with IIT Bombay, with ImmunoAct primarily, of course, but we are also extending it to government and policymakers, and working with industry and international partners and investors and philanthropists to make sure that the whole ecosystem for cell therapy grows in our country. And with that, we also opened the educational initiative to educate and grow the ecosystem so that the workforce for the next generation of CAR T cell therapies and other cell therapies can grow in our country. We have the series of advances in cell therapy events. 3 major events have already been held, and the next one is due in 7th to 9th March 2025. And for the last one, we had more than 550 registrations, and which is showing that the huge interest and scope for these therapies in India. And ultimately, technology must benefit patients. Life comes around a round circle. This is a round circle. This is one of the first few patients treated on the phase 1 trial. She had the opportunity to interact with Emily Whitehead. And I think the question on the top of her mind was that, do the doctors give you IVIG? Which was, so this was a beautiful moment which we were able to capture. And it gives us hope to continue what we do to ensure that girls like Ishwari and Emily continue to, fulfill their dreams. I have a lot of people to acknowledge, but I'll be brief. Hasmukh Jain, of course, is my colleague and dear friend, and he's been leading the adult trials. You're going to be hearing him next. And now Chetan Namne, who's leading the NK cells, Dr. Albina, who's the Chief Scientific Officer in Tata Memorial Center, and this is the rest of my team, including 2 of my PhD students, and in the entire clinical team at Tata Memorial Center. Rahul, of course, who's the bedrock of this whole game and whole story. And Terry, who's no longer in NCI, has moved on from there. And Dr. Nirali Shah and Steve Heifel, who have been so special to us. And the entire Central Global Health team, including Sudha Shivaram, Vidya Vedam, Thomas Cross from earlier, and Fred and Tony. These are remarkable people who helped us with the GMP guidelines. This is to remind you that I'm from the pediatric oncology unit of Tata Memorial Hospital, and I'm happy to have some very great colleagues with whom I'm able to work and do all this. And now I will be handing over to Hasmukh Jain from the adult hematolymphoid group of our hospital, who will talk to you about the adult trials and the commercial railroad and the subsequent journey. So that's it from me, and over to you, Hasmukh.
Thank you, sir. I'll just get started. Right. So I'll be talking the part 2 of this discussion. So I will be now taking forward from where Dr. Gaurav left, focusing on the phase 1, phase 2 registration trial in the adolescent and adult patients, which led to the approval of the CAR T-cell therapy in India. I'd like to first of all thank the Center for Global Health for inviting us to give this talk, and not only this talk, but also the guidance that they've given us in this journey. Right at the beginning in 2015 was the first time when I visited, and that was the first time that this gave me a broad perspective and overview on what CAR T-cell therapy could do. And we are happy that we've reached a point where we now have at least a commercial approval of the product. So this is my conflict of interest. I think the leading reason why this has really come into focus is the fact that this is one of the few therapies where there's a great feeling that this could lead to an improvement in the access, particularly for the LMICs. And this is largely because there's been a dramatic reduction in the cost. The cost of this product has come down to 1/10 of what is the cost of the other products. And so I think this is why this is primarily come into focus. But I think there's a lot of scientific merit also for this development, and I will take you through the clinical data and show how this is important also for us to appreciate. So I'll talk a little bit about Indian context, particularly from the adult hematological malignancies point of view, and go take you to the phase 1, phase 2 trials, talk about the steps that we're taking now on how to improve the access, and also about the future directions. So when we just look at leukemias and lymphomas, there are about 100,000 patients diagnosed per year. And a conservative estimate is that about 20,000 of these patients would require some form of salvage therapy, CAR T-cell therapy being one of them. And as has been told earlier, when it comes to delivering complex therapies that require logistics, that requires trained healthcare personnel, we have been doing this for a while now. And we have 100+ centers who deliver BMT in the country. But in terms of access or in terms of this demand-supply mismatch, there's still a huge gulf. Only less than 5% of the patients who need a transplant actually end up getting a transplant. So things have to be different. And why this is important is because as far as the outcomes in the frontline setting go, we are comparable with the rest of the world. This is the data from our own center in the adolescent and young adult setting, be it leukemia or lymphoma. In the frontline therapy, we are doing all right. The problem happens in the relapse setting where even though the rest of the world has moved on and have got access to some of the new molecules and they've been able to shift the curves a bit, we still represent what would be the standard of care in some of these trials with very dismal outcomes. And for us to change this, we have to have an access. So I think there are 3 issues when we look at options in terms of the in the setting of the salvage setting. You have the molecules, you have the therapies. I think there are 3 issues. One being the access, and the biggest thing is that some of the most important molecules, for example, blinatumomab, glofitamab for lymphoma, tafasitamab, they're not even available in the country. The second issue is about the delivery. Now, auto transplants and allo transplants have been around, but as we've repeatedly said, that there's been an issue in terms of being able to deliver it to all who need them. And I think third in the line would be the cost. So I think even if you are able to first solve the problem of access and delivery, cost is something which can still be taken care of over a period of time. So I think in this context, the development of CAR T-cell therapy as a standalone therapy was very remarkable for us and was very attractive to us. And this is held out in multiple trials, be it lymphoma, be it leukemia, phase 2 as well as phase 3. Consistently, it has been shown that this therapy as a standalone therapy in most instances could cure a significant proportion of these patients. And therefore, this became very attractive to us as a salvage therapy option. But of course, we had to deal with certain problems. So we know that there's going to be an issue with the cost. There's going to be an issue with in terms of manufacturing at large scale, in terms of delivery at the bedside, also in terms of training and getting new centers to deliver this CAR T-cell therapy as we go along. So looking at each of this one by one, let's start with the cost, which is like the most important issue for us. The product cost at the time of launch in the West was $400,000. This is 20 times the cost of an allogeneic transplant in the country. And with such kind of costs, it became really impossible for our patients to access this therapy. And the only solution around this was to innovate locally. So I think this lesson we learned already from our space mission, where they were able to launch this Mars rover at a cost which is lower than the cost of the production cost of this Hollywood movie. So So it was clear that if you want to do high-tech high-tech innovation would require local efforts, and only then you would be able to control the costs. I think that is the first and the most important step that we took in our journey. The second thing which keeps coming up repeatedly now that the cost is so much in focus is that, and it's very important to stress that the lower cost does not mean low quality. Cost is a little complicated subject, and there are a lot of factors which go into the cost. And I was asked this repeatedly in the last meeting at ASGCT on what about the quality of the product, and I was wondering while I was waiting for my return flight back from New York, and I thought I was in a hotel from Marriott chain. I paid $300. I got a reasonably good breakfast for it. But then I thought that closer home, just closer to the place where I live in Mumbai, when I pay $300, what I get for breakfast is this. So it's not necessarily that it always has to do with quality, and you can still do good quality work at lower cost. And this has been already spoken about, that this was the result of the collaboration between some of the premier institutes in the country and outside the country, which led us to this approval. And just in brief, with a focus on what happened in the adolescent and the adult group, is that once the whole preclinical development concluded in 2020, the IND application was made, and the first patient on the adult trial was dosed in 2021, just after the pediatric patient was dosed. We completed the phase 1 trial. We started the phase 2 trial in the end of 2022. By August 23, we had dosed the last patient. We had the early responses data. This data was presented to the regulators. And based upon the understanding that this is a rare indication, this is an unmet need, and the patients would otherwise have dismal outcomes, the CDSCO granted an accelerated approval to this product, and subject to the condition that this has to be followed up with a post-marketing surveillance as well as post-marketing trial, which is something that is being now planned. And subsequently, ImmunoAct, which is founded by Dr. Rahul Purwar, has now taken the lead on how to commercialize this, and 40 plus hospitals have already been onboarded, and they are undergoing training and accreditation so that they could also start doing, and many of these centers have already started delivering CAR T-cell therapy outside the clinical trials. So I'll not go into much of the details of the preclinical work, just to point that this was a humanized CD19 4-1BB CAR T-cell therapy. The advantage of this humanized CAR T-cell therapy, in which played out in the clinical trials and also now in the real world setting, was that right from the beginning, we knew that this is a molecule which is effective, but at the same time had the potential to reduce the toxicities, because the preclinical data showed that it was leading to a lower cytokine production. And even though it was active in the preclinical studies, and this is exactly what we saw in the clinical trials. So our clinical trial process is exactly similar to what most of the other centers in the world would do. So there's an apheresis, the product gets shipped out to the manufacturing unit. Then there is an integrated semi-automated process that they follow under strict CGMP conditions. The process takes about 6 to 9 days for the harvest, till the harvest, followed by QC, and then the product is released. It's cryopreserved, and then it's released based upon the clinical condition of the patient. So overall, we ran the phase 1 trial followed by the phase 2. The phase 1 trial was a single-center trial which enrolled 10 participants. The dose at this point of time was a range. So we said that 10 to the power of 7 till 10 to the power of 9 CAR T cells would be dosed to the patient. The dosing was also done in a fractionated manner right at the beginning. Initially, the patients were lymphoma patients. And as we moved on to the phase 2, this became a multi-center trial. We realized that the activity was being seen at 5 million cells per kg body weight. So this is the dose that we took in the phase 2 trial. And the indication was expanded. So we also enrolled the leukemias as well as the indolent lymphomas in the phase 2 trial. So all in all, 64 patients were enrolled, which predominantly included lymphomas, but we also had 17 patients with leukemia. These patients were across the age range. So the oldest patient on the trial was 71-year-old, and the patients had at least received 2 lines of therapy. The patients who were treated in the trial were truly representative of what we see in the clinic. So a quarter of them had bulky disease, half of them had extranodal disease. In the leukemia cohort, similar to the pediatric trial, the average blast percentage was 61% at the time of enrollment. So this really represented the most difficult to treat patients on our trial. When we look at the flow of the trial, so 64 patients were enrolled. 57 got infused, and 53 got infused with the target dose of 5 million cells per kg body weight. Mind you, the 3 patients were from the phase 1 trial where we had not yet reached the target dose, or we had not identified the target dose. But all in all, when we look at the target dose of 5 million cells per kg body weight, 53 would comprise this efficacy evaluable cohort. And when we look at the responses, it was 70% was the overall response. It was similar in the leukemia and lymphoma. The only difference being that the number of patients with the partial response was slightly higher in the lymphoma cohort. And at the 3-month time point, we noticed that these patients actually progressed. So the complete responders were the ones who truly held on to the responses. And when we look at the swimmers plot, there are a couple of important observations that we have made. One is that the responses are really related to the it's related, one, to the dose that we used, the indication, I mean, the number of lines of therapy that the patient has had received, and also the disease type. So for instance, if you look at this top part of this plot, this is the lymphoma cohort. And if you look at this, that is one line failure, you see more of these blue graphs, which represent complete responses. Whereas the patients who had more than 2 lines of therapy, there was a mixed bag, and particularly some of these patients from the phase 1 trial did not respond well because they'd still not got the target dose. We had not realized the target dose at that point of time. In the leukemia cohort, you see a lot of these patients in the blue, and many of them are still following up. So I think it was the disease type which is important. Leukemias generally tend to do better compared to lymphoma. The number of lines of therapy was also equally important. And third was the dose of CAR T-cell therapy that was used. The toxicity profile is very interesting in the sense that one of the standard features is that there is absence of neurotoxicity, except for one patient who had a neurotoxicity on reinfusion. With the initial therapy, we did not see any patient with neurotoxicity. And in terms of cytokine release syndrome, in the lymphoma cohort, there was no grade 3 or grade 4 cytokine release syndrome. Whereas in the leukemia cohort, it was more or less similar to what is reported with the other products. Another interesting finding was that we had 5 patients who had IEC hemophagocytic syndrome. So that is another one, but most of this was manageable. So just to compare in terms of the toxicities, particularly the toxicities of special interest, that is CRS and neurotoxicity, when you compare with the other approved products, you'll appreciate that cytokine release syndrome is actually much lower for the lymphoma cohort. In terms of the severe CRS, we did not have any patient. Whereas this is expected in about 4 to 20% of the patients, with the other products. And most importantly was the neurotoxicity. So even in the lymphoma cohort, you would expect this to be seen in about 20% of the patients, whereas we did not see any patient who had neurotoxicity. What this meant is that there was a lower utilization of the healthcare resources. So be it the hospitalization, which was short, the average hospitalization being 7 days on the lymphoma cohort and 11 days on the leukemia cohort, including the mandatory hospitalization. It also meant that the need for ICU care was limited. So only 7% of the patients required intensive care unit, besides a reduction in the other important healthcare resources, such as the need for diagnostics, the need for hospitalization for conditioning, etc. Now, this is true on the trial, but what we are seeing, and we're very happy to see that the results have also translated into the real world. So till May, the first patient got treated outside the clinical trial in December 23. This data is till May 24. Till May 24, 77 patients had got apheresed, and now there's an equal split between the lymphomas as well as the leukemia. Out of the 77, 40 patients were from our center alone. And if you see again, there is a similar representation in terms of age, in terms of lines of therapy. In terms of the responses, it is similar, if not slightly better from what we saw in the clinical trials. So we are seeing a response rate close to 80%. And again, in the lymphoma, we have a complete response in the range of 50 to 55%. And in the leukemia, the complete responses are 90% and above. So I think what has changed is the experience and also the better patient selection, which has meant that we have been able to translate the clinical trial results into the real world setting. So this is very important for us because a reduction in toxicity means a lower utilization of healthcare resources. Ultimately, the cost of care goes down, and this therefore can improve the access in the context of our setting. The second important aspect of delivery when it comes to treating patients and doing this, there are 3 important aspects which we were focused right from the beginning: is how to do this safely, how to do this at speed, and how to do it at scale. Because there are patients who cannot wait for the therapy, so you have to deliver the therapy. Your clinics will not go away. Our clinics are as busy as they were even earlier. A regular OPDs in our setting means 200 patients coming in with 5 consultants, a few residents. And so you had to integrate a CAR T-cell therapy program into this. So what we did is we created a manual. So we call this as a pilot manual approach. which takes the patient, which basically records the patient's journey. So it has reminders, it has checklists at every time point in the journey of the patient. So even if a trainee or even if a nurse were to use it, they will be able to make sure that they don't make any mistakes. The other important thing we did was we converted this SOPs into checklists. So we have this lengthy, guidelines coming in. Like for example, the CRS guideline is a big document. We converted this into a simple 5-point checklist. So the patient, so the resident who's treating the patient just follows them step by step. He doesn't have to really know the grading, etc. He just has to follow this, and he will be able to give what is expected of him. So this, I think, has really ensured that we've been able to treat patients safely. The other aspect is at manufacturing level. I think the important decision was whether to go with an automated system or a semi-automated system. So we ended up with a semi-automated system, and the way it works is like an assembly line. So there are 3 different parallel lines which are running. One is doing the plasmid manufacturing, the other one is doing the vector, and then the CAR T-cell manufacturing. It allows for scaling up. It allows for multiple products to be manufactured. It also allows for a better delegation of trained people according to the skill sets to different assembly lines. Now, this is courtesy Dr. Rahul. I'm speaking on his behalf on this slide. So what they have been able to achieve, they have the facility in the western part of the country. What they've been able to do both on the trial and outside the trial is develop robust systems on delivering the products. And they've been able to do this across the country. What they hope to do is to move to something called as a regionally decentralized model, where they'll continue to have the central hub, which, by the way, has currently a production capacity of 500 products per year. And they hope to increase it to 1,500 by the end of next year. But do a regionally decentralized model where some of the terminal steps in the process could be done more closer to the region. Sorry. So I think what we have tried to do is, for the cost, we have been able to reduce the cost by innovation, by developing a product which has a requirement of lower healthcare resources. The production is getting better. This is because of semi-automated model. The delivery is easier because of the careful design and because of the lower adverse event, and also because of the use of a pilot manual. To top it all, we are developing monitoring mechanisms. So we have developed a registry now, which is going to be with the Cancer Consortium, which is going to document all of this from the real world data. We've also just started a virtual tumor board with many centers who log in and discuss the cases. So creating a good forum for discussion in the real time. So once in 2 weeks, we log in and we discuss the challenges that we are facing. And this has again been a great learning experience for all of us. I think at the end of the day, this is all to have success stories. We want many success stories. We want our patients to benefit. So this is just a handful of our patients who have been treated. So some of them are, they're doctors here, they're technicians, they're amateur footballers, there is a cyclist, there is a single child of a parent who are desperate to get the CAR T-cell therapy. There's this oldest, the patient who's crossed 2 years of follow-up, and he just recently had a baby. So there have been several stories around this, and I think this is what keeps us going. So I'd like to conclude by saying that this access cost is a very important issue, and it has a lot to do with the manufacturing, but it also has got to do with in terms of clinical care costs and the ability to reduce those things. So this probably positions Talecaptogen autoleucel, is what it is called, as an ideal CAR T-cell therapy product for LMICs, and potentially has global health implications. So with this, I'd like to conclude and acknowledge several people, our institutes who have helped us wholeheartedly, our collaborators, the regulators really who have kept us on our toes, but also have been helpful throughout our journey, the funding agencies, colleagues and mentors, National Cancer Credo for helping us in developing the protocols, our guides, Dr. Nilarisha is here, Dr. Satyanila, Dr. Nitin Jain, and several others whom even Dr. Gaurav referred to. Most importantly, again, our patients and our families who have placed their trust in this journey. So with this, I'd like to conclude. Thank you so much.
Thank you so much, both of you, for an extremely exciting and engaging presentation. There are so many lessons here to sort of unpack, and I'm glad we have about 12 minutes for questions. We'll now open the session for questions. Please feel free to turn on your cameras if you're comfortable so we can see each other during the discussion. As a reminder, you can type your question in the chat box or use the raise hand feature, etc., and wait to be called on, and unmute yourself if we do call upon you. We'll get to as many questions as we can. I'm going to take sort of the prerogative of being a moderator to ask you a fairly general question, both of you, and I recognize also your colleague who is available, I think, in in the group. One thing I'd like to know is, how has this been received? I mean, clearly the scientific community has been very excited about all the efforts that you've undertaken, and the collaboration alone is a great story between a high income and a middle income country. But have you had any pushback? Have you found it hard to convince others, for example, of how great things can be when great minds come together, regardless of the country? And a related note about the pragmatic issues in terms of cost. I see there's quite a few questions about cost and technology transfer also in the chat. So who would like to go first if you want to address this? Dr. Jain or Dr. Narula?
Yeah, I can. I mean, I can start with. So I think in terms of pushback, I don't think there's been pushback. In fact, we've been fortunate that we work in Tata Memorial Center, and the support for this whole journey has been very. It's been remarkable. So I think that in terms of scientific community, again in India, as well as the clinical community in India, they've been very like excited about this development. As I said, 40 plus hospitals have already signed up for this to be a part of this. They have been like getting the training, etc. Some of them have already started. There is a very good amount of discussion going around within the community. So I think that I don't think has been a problem. Yeah, I think. What is the second part of your question? Sorry, this missed. The cost, is it?
Technology transfer and also cost. A few people have asked in the chat, so I thought I would just throw that in directly.
So the cost of the product right now is about $40,000. Yeah, it's about $40,000 for the product itself. I think the technology transfer, I leave it to Dr. Rahul to answer.
But the cost then would be 1/10 of what we'd have in the US. Is that right?
Yes, it's $40,000 right now. Yeah, about $40 to $50,000.
Dr. Pawar, would you like to respond to any of that as well?
Yeah, no, thank you so much. I think cost has been, I think, a very good factor for the access in our country. And I think beyond the cost, I think what Dr. Hasmukh and Dr. Gaurav sir has mentioned, the quality of the product, even at that price point, the quality of the product in the sense of efficacy as well as toxicity profile. I think that's what making a difference in providing the access to the patient. So far, we have treated now almost 150 patients are already been infused, and around 200 patients are offered the CAR-T. And like every month, we are treating a lot of patients. So I think this really making a lot of difference. In fact, today we have heard the news that one patient has become father. Like he has been post-CAR-T 2 years. He's been like in the complete response, and he just became the father. So this is like really motivational, that inspiring, that we have to continue our hard work and ensure the access of the CAR-T.
Thank you so much. Dr. Narula, did you want to add to that or respond to a different question? We have a few coming in.
No, I think it's well covered. Just to say that from the earlier stages onwards, I think we never faced any pushback. I think probably institutional, reputations matter. The fact that it was Tata Memorial Center and IIT Bombay, and we were collaborating, with you. I don't think anybody from the scientific or the clinical community ever doubted. We received support all along. I think we only faced difficulty initially from grant agencies, which was skeptical, which is understandable. In retrospect, I forgive them, so to say.
I'm going to say you showed them. Yeah, great. And I see a few questions that are more on the technical side, including one from Dr. Ramila Shilpakar, who I know. What is the outcome of patients with CRS, that's cytokine release syndrome? I think you addressed that to some degree towards the end, but who would like to address that? Thank you. Dr. Jain, perhaps?
Yeah, so in terms of cytokine release syndrome, so we've seen like complete recovery from cytokine release syndrome. Most of it was grade 1, grade 2. The average requirement of tocilizumab was 1 dose, except for a few patients who had a grade 3, grade 4, and those were the patients from the leukemia cohort when we were treating them early on in the trial with active, rapidly proliferating disease burden. Now, having moved on to the real world, where we really don't get, we get such patients, but then we've learned our lessons, and we give therapies to get the disease burden down. And from that time on, I mean, at least in the real world setting, we have not seen any grade 3, grade 4 CRS even in the leukemia cohort. So yeah, so we've not had a CRS related mortality on the trial as well as subsequently.
Right, and a related. Oh, please go ahead, Dr. Narula.
No, sorry, I would agree with that. So because the pediatric trial is almost exclusively leukemia, and we have treated patients with very high burden disease. But even in that case, we have not had anything beyond grade 2 CRS. The solitary patient being one who had multiple host-related factors, and which led to it rather than anything else. So I would concur with that. It seems to be remarkably low toxicity. We were preparing for the worst while doing this trial, but so far we have not had to encounter that situation.
I think a lot of people will be very interested in the longer term outcomes, and we heard that one participant is now father, which is great. But somebody was asking about 1 and 2 year mortality. I thought you'd already had that. How much? Where are we at with follow up? Yeah.
Yeah. So we are on for the adult trial, for example, we just dosed, as I said, the last patient in August 23. So we are now waiting for at least 12 months of follow up for the 12 months of follow up after the last patient got dosed. And so we are in the process of actually putting together a manuscript, and then we'll soon be reporting the reasonably long term data very soon. Yeah.
The earliest phase 1 trial participants are only now crossing the 2 year landmark point. So I think that data will grow only with time. But we have 2 year survivals now, or long-term remissions now, including the girl which I showed you. Yes, so she's she's out 2 years now.
Yeah. Unless one of you also want to respond, Dr. Satish Gopal, our director, has the last question. Please go ahead, Satish.
Sorry, thanks, Sophia. I didn't mean to take the last question, but thanks to all of you for joining us today. It's been really wonderful, I think, to get to hear an overview of the entire story. And I know it's one that the NCI feels really proud to have contributed to in a small way. I have a couple of questions about kind of where you see this work going forward. Because there have been so many questions about the cost in the chat, I'm curious on the engineering side, how much lower you all think that you can drive the costs. I'm not asking you to commit to a number, but I think as you scale this experience, it would really be interesting to kind of hear from you. Do you think you have plateaued where the cost, this is sort of what the cost will be in India, or do you think that there's an opportunity to drive that much lower? So that's one question. Also, less on the engineering side and more on the science side going forward. What you've done, I think, is replicate a product that a product targeting an antigen that we knew worked from high income countries, right? And you've done that at a much lower cost and within India, which is critically important. I would never devalue how important that is, particularly as someone who's worked in LMICs for my entire academic career. I'm curious if you're also interested in exploring new antigens that may be unique to India or that haven't been well characterized in other settings. So both on the engineering and the science side, I'm curious about sort of what you think the cost horizon might be, and then also what you're thinking about in terms of antigen targeting going forward.
Yeah. Can I answer this question? Yeah.
Go ahead.
Yeah. So regarding the cost, I think it will further come down as we scale up, because the facility we have created, and we can clearly see in that model that as soon as we scale up further, the economics would be better and we can further reduce the cost. So as you know, any technology, whenever it comes, they are expensive. And with time, once the technology matures, the prices goes down. I think similar trend will happen in our country as well. And today we have this lentiviral vector-based CAR-T. Lentiviral vector is very expensive, right? So I'm sure even those platforms would evolve, and then we will have further cost down. And regarding your second question, in fact, I would request NCI, NIS to continue with our great collaboration, and maybe we can work together and take the new antigens and novel antigens and do the clinical trial here. Because one of the, I think, biggest success of our collaboration is that we have created an ecosystem in India to do the translational work and to do the clinical trial. And that's what can be now leveraged by the NCI scientists. And now we can even have more and more collaborations where we can do the early clinical trial or the latest stage clinical trial, whatever you are comfortable with, and get the results very quickly, because we have a lot of patients in this part of the world, and all these things can be tested because because of this collaboration of NCI, IIT Bombay, Tata Hospital with Dr. Gaurav sir and Dr. Hasmukh, we have a very good ecosystem.
That is very exciting. And what a way to end. What a way to wrap up this excellent seminar today. Thank you so much. That, I'm sorry to say, concludes our session today. Very special thank you to Dr. Narula, Dr. Jain, and also Dr. Purwar for contributing. And remember to join us for our next session on August 8th with Dr. Martin Lajou, where he'll discuss cancer control and implementation science in Mexico. And once again, please visit our website, cancer.gov/global health, to find out more information about upcoming seminars, past recorded sessions, funding opportunities, events, news, and more. And follow us on X at NCI Global Health and LinkedIn. Thanks very much.
Thank you.
Thank you. Thank you very much.
Thank you.
Bye-bye.
Thank you.
Thanks
Bye-bye.
so much. Bye-bye.
Thanks, Sudha. Bye.
U.S. Department of Health and Human Services, National Institutes of Health, National Cancer Institute, cancer.gov, 1-800-4-CANCER.